talk to you about protein. You now know all about carbs and lipids, at least you did before the break, but that's not a problem because I'll make sure to remind you of everything that's relevant to study this third group of macronutrients, which is protein. Before we start, I would like to bring up, this is horrible. I would like to bring up three misconceptions that are quite common about protein, and that I'm sure that the not so nutritionally educated family here, the fat family on the slide, would probably share. The first one, that protein's the good stuff. A lot of people who do not have a lot of knowledge of nutrition have this concept on the back of their mind that protein is somehow what you really should be looking for in food when you eat, like the stuff you need to grow strong and maintain your tissues. And that is of course true, but now of course you know better. You know that carbs and fats are equally as important. They are important energetic molecules. We need them also for structural functions and also for regulatory functions. The essential fatty acids, we cannot survive without them. So all three, the macronutrients, are vital, are very important for us, not only protein. The other one is that protein is animal, meaning animal food is the best, somehow, source of protein in our diet. And again, this is not completely true, because plants are also very good sources of protein, which is the reason why the lean family in our previous slide was able to perfectly thrive on their vegan diet. And then the last one is protein makes muscle. Well, protein does not. Exercise makes muscle. Protein is a component of muscle. We need a little bit more protein if we want to build new muscle, but certainly eating protein alone doesn't have by itself any kind of anabolizing effect. It's like you wouldn't throw there a bunch of bricks and think you're building a house. You still have to do it. If you just eat extra protein and do nothing else, it will just convert this protein to fat and get fat, and certainly not athletic. Okay, this is weird. Well, let's look at the top one. Proteins are the main structural material in our body. 62% of our body weight is water, thereby making water the most important structural material in our bodies, what really keeps us in shape. But then the second most abundant structural material is protein. 17% of our body weight is protein. So our muscles, our bones, our skins, all of our organs, our hair, our nails, protein is the stuff all these things are made of. But then on top of that, proteins also have key regulatory functions in our body. A lot of hormones, all the enzymes, the carriers, the transporters, the signaling molecules, the antibodies for immune function, and we will go into detail of all this function later. But this is just to say that proteins are vital to a lot of metabolic processes. So they not only give us structure, but they are also what make all this structure work. And then, of course, I have no clue what's going on, but now we look at the third. Protein can also be used for energy. So sure enough, they provide 4 kilocalories per gram, so just about like carbs, if you remember. And actually, protein will eventually give you a little bit less, but they can be used for energy, although using protein for energy is kind of a waste. Our body doesn't really like to use protein for energy because they are normally used for other more important structural and regulatory functions. Yes, we know that. Now, what a protein is a chain of amino acids that are linked together by peptide bonds. So a protein, you can imagine it as a linear chain of these single units, these building blocks that are the amino acids that are linked together by this type of bond that I will show you later. And so they will form this linear sort of necklace of amino acid that will then fold into the three-dimensional space to acquire its characteristic form, its characteristic shape. That is what most of the time will give protein its biological functions. But if any time we take a protein and unfold it, we will end up with a linear chain of amino acids linked together one after the other. So amino acids are the building blocks of proteins, and our body needs to build thousands of different proteins for all its structural and regulatory functions. But all these thousands of different proteins are made with only 20 different amino acids. So the basic building blocks we need to build all the thousands of proteins that we may need to build is only the 20 amino acids. And by combining them in different combinations or in different numbers, because a protein can be made of 3 amino acids or can be made of thousands of amino acids in sequence. And then we can build all the proteins we need with these 20 basic building blocks. So you can imagine that what we really need when we eat protein from food is not really the protein that we are after. The protein we eat from our plants, our animals, they will have their specific biological functions in the plants or the animal that they come from, but we don't care about the function. We only care about the pieces, so we care about the amino acids. So we will take that protein, as you can see here, we eat protein from foodstuff. These little circles will represent the amino acids. You see they are linked together to form this chain, which is the protein with its function. We don't care about the function. We just break it down into the single amino acids that make that protein, so that then we can take the amino acids and reassemble them together to form other proteins. The proteins that we need with the functions we need, that is not anymore related in any way to the original function of the protein we ate. We only are after the pieces so that we can build the protein we need. And now, here we represent the amino acids as this little circle. How an amino acid really look like is more of like this. You can recognize that there is a central atom of carbon that, like any good central atom of carbon, will make 4 bonds. One is made with hydrogen here. Another is made with this carboxyl group, is an acid group, carbon, double bond oxygen, but hydrogen, that you're already familiar with this because it's the same carboxyl group that you have at the terminal end of your fatty acids. It's what makes them acidic. But then we have a little surprise here. We have an amino group, which is nitrogen, hydrogen, hydrogen. And if you think back to your carbohydrates and lipids, there are no nitrogen in carbs and fats. So protein are our only, well, not our only, but our main source of nitrogen, which we need. So on top of carbon and oxygen and hydrogen, amino acids also bring us nitrogen. And then this R here is not really an element. It's just a shorthand for a side group that will vary and will determine the specific amino acid. So this bottom part here is the same for all our 20 different amino acids. And then what will change is this side group. So for example, if the side group is just an atom of hydrogen, we will have made up the simplest amino acid, which is glycine. If instead of hydrogen we put there a methyl group, carbon, hydrogen, 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 that's alanine. And so on, you can build all the 20 different amino acids by changing that side group. So this is an amino acid, but then we said, all right, now we want to make a protein, and a protein is a chain of amino acids linked together, so now we need to bind them, and we do that with a peptide bond that works like that. You can see here there is 2 amino acids. You will recognize the carboxyl group here, and then the amino group of the other. The peptide bonds will happen through elimination of a molecule of water. We take oxygen, hydrogen, hydrogen from these groups, so this is a condensation reaction. Water will leave, and then we will form a covalent bond between the carbon of the carboxyl group of an amino acid and the nitrogen of the amino group of the other, so they will share their pair of electrons, and now they are bound. And of course, each amino acid can make 2 of these bonds, one on one side and one on the other. So for example, this nitrogen of this amino group will be able to bind to the carbon of a carboxyl group of another amino acid, and this carbon of the carboxyl group will be able to bind to a nitrogen of an amino group of yet another amino acid, and this way we can build the chain as long as we want. Thousands of amino acids if needed. This process of building proteins from amino acids is what we call protein synthesis, and this happens in our cells. This picture is the one of a cell. Now I quickly go through how it happens. Of course, those of you who have studied biochemistry already know this more in detail, but here we just summarize how it is. And then on the next slide, I'll write a few sentences down to summarize again the same concept. So this is a cell. You can recognize there is a nucleus, and then outside the nucleus there is a cytoplasm, still inside the cell, but outside of the nucleus. And inside the nucleus we have DNA, which contains the information that's necessary to make our protein. So when we need to build a protein, suppose we need to make insulin, so now we would be in the pancreas, we first go and look for the genetic instruction, the code, the recipe to make insulin in our DNA, which tells us what is the sequence of amino acids that we need to put together to end up with that specific protein. So we open up this sort of recipe book at the gene of insulin that is written in DNA language, so it has a sequence of nucleotides that will then be translated, but basically say, okay, this is the sequence you need to make insulin. And because this is very precious molecules, it's a precious recipe book, it will not go out of the nucleus to the cytoplasm for transcription. It will first be copied into another molecule, the messenger RNA. So we will first make a copy of the instructions. In actually, we do not only make one, we make many. Imagine you take this recipe book and you make many photocopies of it. And then this can safely go out through these pores in the nucleus into the cytoplasm to these other structures that you already know, the ribosomes. And if DNA is the recipe book, then the ribosomes are the cook that will take this recipe, will take the mRNA, read it, and start building the protein following the instructions. But of course, this is not as easy because, as I said, the recipe for protein in mRNA is written in a different language. So it also needs to be translated into protein language. And what does that is this other molecule here, the transferred RNA, the tRNA, that will be able to tell, okay, this is the nucleotide here. This means you need to take this amino acid. So we'll take the amino acid, we'll make a peptide bond with the previous one on the chain, and you will end up with this chain, which again would be a sequence of amino acid. And then on to the next one. Here we have the instruction, 3 nucleotides. The tRNA will say, all right, this means take this amino acid, we will take it, bind it to the previous one, and so on. This chain of amino acid will grow. All right, and this is how we make protein. And because many ribosomes are working on the mRNA at the same time, this will go very fast. So this will basically say the same thing. Yeah, I'm sorry, it's a lot of stuff, but it's the same thing I just said. DNA in the nucleus contains the coded instructions to make our proteins. These instructions will be copied to mRNA, and then this will be able to exit the nucleus and be transferred to the cytoplasm. Ribosomes in the cytoplasm will then read this mRNA and start building proteins by adding one amino acid at a time. The tRNA, transferred RNA, will provide the translation between the DNA language, which is the sequence of nucleotides, and protein language, which is the sequence of amino acids that our protein is made of. And so this is where the actual translation will happen. And now here's a new concept. The sequence of amino acid in the protein will determine its final shape. So this linear chain will then fold, we said, in the 3-dimensional space to have a specific shape. But the way it does that is not random. It is actually determined from the linear chain of amino acid itself. So once we have the linear chain of amino acid, there's only really one possible way that it will fold in the 3-dimensional space. And depends on the sequence of amino acids, the way they will interact. And this is very important because most of the proteins work only because they have a specific shape. So the final shape will determine the biological function of the protein. But this shape is direct consequence of the sequence of amino acid of the linear chain just as they come off of the ribosomes. And to illustrate how this is important, in your book there is this example of sickle cell anemia. That is a genetic disease whereby you have a little mistake in the recipe for hemoglobin. So in your DNA, the gene encoding for hemoglobin has a little mistake that will result in a wrong amino acid in the chain of this protein. And hemoglobin, as you know, is an important protein for your red blood cells so that they can then efficiently carry oxygen. This mistake will result in the fact that in position 6 of the chain of this protein, you will have valine instead of glutamate. So one amino acid instead of the other. But this is enough that the protein will then fold three-dimensionally in a completely different way, which is dysfunctional. And because the shape is what determines the function of a protein, this will result in a totally dysfunctional red blood cell itself. So you can see the whole structure of the red blood cell. This is not the protein, this is the whole red blood cell. Will have a completely weird shape that is not as efficient in carrying oxygen. And this depends on the wrong shape of hemoglobin, which depends on just one little amino acid put in place of the other. So you see how important it is that the sequence of amino acids is the right one, and how this then determines the final shape of the protein, and how the final shape is important for the function of the protein itself. And so you can imagine that if a protein loses its three-dimensional shape, it will also lose its biological function. The loss of three-dimensional structure of a protein is what we call protein denaturation. So it is basically the unfolding of the protein back to a wrong shape or to its original linear shape. The linear shape will be conserved, but the function is now lost. And this can be something that we do not want, for example, for the protein in our body. Our insulin that we just built, we don't want it to be denatured because then it will lose its function. But in other circumstances, this would be something we actually want. Think protein digestion. We said before, what we care about digestion is not preserving the function. We don't care about the function. We just want the pieces. And so the denaturation of the protein now will be something we want, because it will just make it easier to unfold the protein and then start breaking it down to get the single amino acids that then we can use. So one of the goals of protein digestion will actually be protein denaturation. And there are many ways that you can accomplish protein denaturation. These are the most important. Heat is the most important way you can accomplish protein denaturation. So you have here your egg with a transparent and liquid part. If you cook it, it will become white and solid, and this is irreversible. If you cool it down, it will not go back to transparent. And this is a result of denaturation of the proteins of the egg white induced by heat. And in general, every time you cook a food that contains protein, you will, to some extent, have some protein denaturation, which is in general good because it will then make digestion of that food easier. Another way you can do that is with enzymes, and again, think digestion. Some enzymes will actually help unfolding the protein to accomplish more efficient digestion. We can denature a protein with high or low pH solution. Again, think digestion. In your stomach, there is a very acidic environment that will totally help protein denaturation. Or, I don't know if you've ever squeezed some lemon juice in milk, and you will immediately see protein precipitate. That's kappa-casein that is partially denaturated by the effect of the acidity of lemon juice. And also agitation, which you also have to some extent in digestion. If you think of that shaking, that is basically to move things on and to help lipid digestion, but it also to some extent will help unfold proteins. Or if you've ever tried to make mayonnaise, You know that you have to agitate your egg yolk so that you can then denature the protein and incorporate your oil, because if you do not do that, it will just end up with a sort of disgusting mixture of egg yolk and oil. On to the next concept of essential amino acids. One amino acid, as you probably already have realized, cannot substitute for another in protein synthesis. We'll see how the code for building a protein is in very detail written in our DNA. We cannot just take another amino acid and replace it. Did you see what happened in sickle cell anemia? You just replace one and you mess up everything. Luckily, however, of the 20 amino acids that we need to build all our different proteins, 11 can be interconverted into one another. So, and this is the transamination reaction. Meaning, if we need a specific amino acid to build a specific protein and we do not have it, in most cases we can take another, and using stuff that's usually readily available around the cell, we can convert it into another amino acid which we can then use. However, we cannot do that with the remaining 9 essential amino acids, and we call them essential just because of that, because we cannot make them ourselves. These 9 amino acids cannot be interconverted or made starting from something else, and so they must come directly from food. And we need all of them, because if we miss just one, then we will be in trouble when we need it. And this is a table you have in your book showing the different essential amino acids. I don't have to remember names, but just the concept that these 9 essential amino acids are the ones that we cannot build ourselves, and the other 11 can be somehow interconverted into one another. And to illustrate again this fact, I will bring another little example. Imagine the cell needs to build this imaginary simple protein. It's just a 3 peptide, so made of 3 amino acids, glycine, aspartate, and lysine. And so what will happen is we'll look for the instructions in the DNA, make our RNA, bring it to the cytoplasm, give it to the ribosome that will start reading it and say, okay, so to make this protein we need glycine. And imagine this is the pool of amino acids that's available in that specific cell at that specific moment. We need glycine, here's glycine, so we can take it, that's good. And on to the next one, now we need aspartate, but we do not have any aspartate. However, it turns out aspartate is one of the non-essential amino acids, and it can be made from glutamate. So what we will do is we can take glutamate, make a transamination to aspartate, and now we're good to go. We can take aspartate, bring it next to the glycine, make a covalent bond, our peptide bond, and on to the next one, now we need lysine. Again, there's no lysine available, but now we are in trouble because lysine is one of the 9 essential amino acids. So there's no way we can make lysine starting from what we have. It doesn't matter. We can have thousands of other amino acids. We will be stuck, and we will not be able to make the protein we need because lysine is essential. We don't have it. It doesn't matter how much protein we have unless we have that amino acid. We are stuck. We cannot go on with our protein synthesis, which, of course, is not good. So this brings us to the so-called all or none principle, which says depletion of just one essential amino acid prevents protein synthesis. We do not have lysine. There's no way we can make our protein using something else. And so either all essential amino acids are available or none can be used. It doesn't matter that we have a lot of the other ones. We want that one that we need. And then on top of that, the remaining amino acid cannot even be stored for later use because our body doesn't really have any system to store proteins or amino acids. So we cannot even say, all right, we don't have lysine, we will just save this and for later, and then once we have lysine, we bring them back and make our proteins. No, it doesn't happen. We cannot store them, so if we have no use for them at that specific moment, we will have to throw them away or use them for something else, but certainly not for protein synthesis, which is the most important use we have for amino acid. And so you see the quality of a protein, food, or protein source, depends on its essential amino acid content. Our protein requirement will require a total amount of protein, like how many grams of protein we need. But it also will have a requirement for quality. So we want to make sure that we have all the 9 essential amino acids, because if we don't have all of them, then we can have all the protein we need. We will have no use for it, for protein synthesis, if we lack just one of the essential amino acids we need. And so there is this example, back to the beginning of the 19th century, this French scientist, François Magendie. Back then they already had these early nutrition studies, and they knew that protein was very important. And actually back then they thought that protein was the only thing you really needed. But they also knew that protein sources, the richest protein sources, were very expensive. There was, however, this food, gelatine, that is an animal source of protein, a lot of protein, 86% of protein per dry weight. I don't know if you're familiar on how gelatin is made. It's not the best thought. It's made from collagen. So basically we'll take bones and skins of animals, dairy, chicken, fish. You will boil this for a very, very long time, and you will end up with this sticky thing that you can then dry and get this powder that has a very nice property. If you heat it, it will form a very nice, tasteless, reversible gel with water. Reversible meaning you can heat it up, it will become liquid, you cool it down, it will become solid, and you can do this over and over again. And so the food industry really likes that, because to make, I suppose, ready-to-eat food or sauces or meat products. And of course, if you put some sugar and colors and flavor, you make your gel or dessert. But for François Magendie, this was the solution to the hunger problem. He said, this is a rich source of protein, it's very cheap, it must be very good. So he gave it to a group of dogs, expecting that they would thrive, and instead they would die, against every reasonable expectation. That's what he said. But today we know that collagen, yes, it is a very rich source of protein, but it is a very, very low quality protein, because it lacks not 1, but 4 of the essential amino acids: tryptophan, isoleucine, threonine, methionine. So it doesn't matter that you have a lot of protein from gelatin. It's protein that you cannot use because you will lack 4 of the important essential amino acids. So high protein, low quality. This is a typical example to illustrate how protein quality is as important as the quantity. And then another little concept, the one of conditionally essential amino acid. These are amino acids that are not strictly essential, meaning we can build them ourselves, but we can only build them starting from essential amino acid that may be lacking. Maybe we don't have enough, maybe we're using a lot. So if that then become lacking as well. Take tyrosine. It is conditionally essential because we can make tyrosine from phenylalanine, but phenylalanine is essential, so if we lack phenylalanine, we will lack tyrosine as well. Phenylketonuria patient, they lack the enzyme to make this conversion. This is another genetic defect. Those people will not be able to take phenylalanine and make tyrosine, and this will result in 2 different problems. The first one is, as you can expect, tyrosine then becomes essential, because now you cannot make it from anything else. And tyrosine is also very important because the metabolism of phenylalanine will then lead to the production of dopamine and acetylcholine, epinephrine, a bunch of important hormones and neurotransmitters. But also, they will have to be very careful and strictly control their phenylalanine intake, because if they will have excess of it, they cannot metabolize it, they will accumulate it, and phenylalanine is neurotoxic. So this would result in serious neurological damage, which is the reason why, if you remember when you studied your artificial sweeteners, whenever a food contains aspartame, it has to carry the warning, this is a source of phenylalanine, so PKU patients beware. And then, of course, these people know, because all newborns are tested for PKU, so that then their diet can be strictly controlled. We see the sources of protein in our diet. animal food has this this is why it's very clear this is a good source of protein, because it has a lot of protein and it also has a very high protein quality. It has a lot of all the essential amino acids we need. And if you think about it, because our requirement for essential amino acids is basically how our body is made. And so you can expect that animal food will have a composition similar to ourselves. Yeah, ideally, human meat would be the highest protein quality you can expect, but of course it's not polite to eat your roommate for lunch. So then you turn to other animals. And here you have some numbers. You don't have to write down the numbers, of course. It's just you have an idea. This is percent weight. So that means 100 grams of egg have 13 grams of protein. Quite a lot. Meat, 15 to 20%. Fish, about the same. Tuna has a little bit more. Milk, yogurt, 3%. It doesn't look much, but keep in mind that milk is primarily water. So if you see how much of that energy comes from protein, that's quite a lot. Cheese concentrates that protein 25 to 35%, and they also are highly digestible, so good sources of protein, of course. But, like we said at the beginning, not the only ones. Nuts and seeds, for example, from the vegetable world, are very good sources of protein. They have a lot of protein. Again, just some examples, peanuts. Well, peanuts is technically a legume, but nutritionally, we don't care. Walnuts, 24%, sunflower seeds, it's the same seeds. They are good sources of protein, and they also have a pretty good protein quality. Not as good as the animal food. They could use a touch of lysine more, but overall it's good. However, these are very energy-dense foods, so you can only eat so much, which is the reason then why you will then turn to, for example, whole grain cereals. And they also, if they are whole, have quite a lot of protein. Not as much, as you can see, as nuts and seeds, but of course you will eat more. So a serving of pasta is, of course, bigger than a serving of sunflower seeds. And they also have a composition that's surprisingly close to the animal sources. However, they have one big problem. They lack one essential amino acid. They have only 1/3 of the lysine. But because we said if you just miss one, you're in trouble, you cannot use the other, this dramatically lowers their quality. So cereals by themselves have very low protein, fairly low protein quality. There are the pseudo-cereals here, quinoa, amaranth. They're not technically cereals, but they are nutritionally close. However, they have a little bit more of this lysine. So they have a slightly better protein quality. And then we also have legumes. Lentils, chickpeas, soybeans, 36%. That ends up being even more than meat. But they also have a low protein quality because now they miss the sulfur-containing essential amino acids. Well, methionine is essential. Cysteine is another of those conditionally essential. But this also is enough to lower their protein quality because of this lack of this essential amino acid. And soy product, again, they have much higher sulfur-related amino acid content. So they have a slightly higher protein quality. We then have fruits and vegetables. They have good protein quality, but they have very low protein amount. So fruit is very, very low. Vegetable is slightly higher, but still less than 3%. So it certainly contributes to your protein requirement, but you could not get all the protein you need from vegetables alone. You would need to eat tons. I definitely can go to the previous slide.
We go to the one before that?
I can go to the first one. So far, so good? And finally, we also have some alternative sources of protein. You have here tofu from soy, tempeh from soy. Corn is made from fungus, so it produces a lot of fairly good quality protein. Seitan is made from gluten, so from whole grains, the protein part of grain. Whey protein from milk, this is rich in branched chain amino acid. And then, yeah, remember that there are, in other parts of the world, people that will eat insects. So this is mealworms, they are animals, so a rich source of cheap protein. This is a picture from a Vietnamese market with bugs and larvae and insects. Good sources of protein, cheap. And tasteless. I don't know if you've ever tasted a bug. I did. A dry one, it was crispy with salt. It tasted salt and nothing else. So it's just, once you're past the psychological barrier, yeah, that's fine. The last concept I'd like to bring out today is the one of complementary. Is this working? I don't even think it's working. Complementary proteins. So the protein quality of a single food. Now we said, all right, these are different food. This is a high quality, this is low quality. But you want to keep in mind that when you eat, you don't really care about the protein quality of a single food. What you care is about the protein quality of the meal, so the combination of food that you eat. And so 2 or more lower quality protein sources, if you eat them at the same meal, not necessarily the same dish, but at the same meal, certainly not one at lunch or one at dinner, because remember, you cannot store amino acids for later use. But if you eat them together, they can then compensate for essential amino acid deficiencies and thereby result in high quality protein meal. And I have another little example to illustrate. Oh, sorry. All right. So imagine again we want to build the same protein. This time I don't even have the names. I just have this free peptide made from a green amino acid, an orange amino acid, and a blue amino acid. Imagine we have this food 1, that has 30 amino acids, 10 green, 10 orange, 10 blue. How many protein can we build with this pool of amino acids? Well, of course, we can build 10, right? Because we take 1, 1, and 1 for 10 times. So we can make 10 molecules of the protein that we need. So we could say that the protein value of this food is 10. This food is a high-quality protein food because it has all the amino acids that we need in just the right proportion. We'll be able to use all of them and make the protein that we need. Now imagine we have this other food that now has 10 green, 18 orange, and 2 blue. And notice that the total protein content is the same. It is still 30 amino acids. So the grams of protein would be the same. But now how many of our protein can we make? Well, of course, we can only make 2. Because after that, we will be out of blue, and we will not be able to build any other of the protein that we need. So we can build 2, and blue is the limiting amino acid. We don't have enough to go on building. And we will have, because we cannot store them, remember, we will have to get rid of 8 green and 16 orange. So the protein value of this food is much lower, is only 2. We can only make 2 proteins. Same amount of protein, but lower quality. And also, let's now take this other food 3. Now it has a different combination, 10 green, 2 orange, 18 blue. Again, we can only make 2 protein, because then this time the orange will be limiting, and we will have to throw away 8 green and 16 blue. And so the protein value of this food is also only 2. But now imagine that we take food 2 and food 3 together. I halved the amount because I want the total amount of protein to be the same. So again, we only have 30 amino acids total, but now the lack of orange in one will be compensated by the other, and vice versa for the blue. So how many protein can we now build? We have 10 green, 10 orange, 10 blue. We can again see how they complement each other. They kind of mutually integrate each other. We can again build 10 of our proteins. We don't have anything to discard. So the protein value of the combination of these two foods is now 10, which is more than just the sum of the individual food. If I eat one at lunch and one at dinner, I will have 2 and 2, 4 for protein quality. But if I eat them together, because of the way they integrate each other, then I have a much higher protein content. So it's 2 low-quality food by eating them together, we make a high protein quality meal. And the most important practical application of this concept is cereals and legumes. They are the typical example of complementary proteins. Because we said cereals, all right, they're good, but they lack lysine, but they have a lot of methionine and cysteine. And then legumes then they have low methionine and cysteine, but then they have high lysine. If we eat them together, they complement each other and provide all the essential amino acids. So if you combine them in the right proportion, which is about 2/3 cereals and 1/3 legumes, now you have the same protein quality that you would have with animal protein. So if I eat my chickpeas hummus on a slice of whole grain bread, then I have complement. And finally, I like to show you the way your government wants you to eat now. This is a new plate, which is the evolution of my pyramid, which I think is like in your book, which is the evolution of the food pyramid, which was, I think, more intuitive. It is, for protein, as you can see, it's kind of weird. Aside from the fact that you have this dish, that to my Italian side, I wouldn't want my pasta with sauce anywhere near my strawberries, but the thing is, protein is not a food. Protein is a nutrient. So you have dairy here, which I assume to be milk, since it appears to be in a glass. But if it is cheese, then it would have to go here. It would be a protein source. You don't want to eat meal and cheese together. And what about grains? Grains are also, and now it's late, sources of protein. Thank you. So I can say goodbye and everybody can hear me. So you see, yeah, it's kind of weird. I think that unless you read the explanation, most people would infer that this is animal protein. And then grain, what did it cost to put a whole grain? Because it changes everything. If it's refined, it's just sugar. It's starch, but your enzyme will degrade it so fast that it's just as if you were eating sugar straight from the sugar bowl. But okay, so this is it. At least it gives you the message that this is the amount of protein you need in your food. And thank you for your attention. I'll see you on Wednesday. You can hear me from back there. Yeah, I think the oxygen sound is good.